Welcome to the Sand Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. Before we jump into today's deep dive, please take a moment to like, subscribe, and share this with your friends and family. It really supports the Sand Science Experience and helps us keep unpacking this incredible research. So today we're tackling something really fundamental in synthetic biology. A really core challenge. How do you power an artificial cell? You can build these amazing microscopic machines, but, well, they need energy. They need a battery, basically. And getting them to generate their own power efficiently and stably, that's been a huge roadblock.
It really has because, in nature, every living cell has its powerhouse, the mitochondria. That's what makes ATP, adenosine triphosphate. It's the energy currency for everything the cell does. But the really tricky part, the bit that's been so hard to replicate from the bottom up, isn't just making ATP, it's setting up and maintaining that stable proton gradient across a membrane. That proton motive force, that's the engine.
Exactly. Getting that gradient stable has been, well, elusive, but maybe not anymore. We're looking at a fantastic paper from Anjuante Chemi International Edition. It came out relatively recently, detailing how they built this mitochondria mimicking ATP nanogenerator. So our mission today is to unpack how they did it. Specifically, this really clever self-amplifying system they designed, and crucially, how they got it to actually power metabolic processes inside an artificial cell model. Okay, let's dig into the core first. This proton generator they call GC at SiO2. Sounds a bit technical, but what's it made of, and what problem was it designed to solve?
Right, so the core components are two enzymes, glucose oxidase, or GOX, and Catalase, CAT. Now, GOX is great for generating protons. It reacts with glucose, lowers the pH. That's what you want for a proton gradient. The big problem, though, is that GOX also makes hydrogen peroxide, H2O2.
Ah, the byproduct issue. And H2O2 is pretty nasty stuff for biological systems, right? Cytotoxic.
Exactly. It builds up, it damages things, it basically poisons the system you're trying to build. That's been a major headache for GOX-based energy systems.
So you've got an engine, but it spews out toxic exhaust that shuts itself down. How did they get around that?
Through really clever packaging. They use this process, an interfacial confined sol gel method in a mini-emulsion. It sounds complex, but basically they managed to trap both GOX and skate together inside these tiny silicon nanocapsules, the SO2 part. And they did it really efficiently, like over 97% encapsulation. The GOX and skate are forced to be neighbors, essentially locked in the same tiny room.
Okay, trapping them together. So the poison producer and the cleanup crew are right next to each other. And this leads to that self-reinforcing cascade you mentioned. This sounds like the really neat part.
It is. Because here's what happens. GOX does its thing, produces protons good and H2O2 bad. But because CK is right there, it immediately grabs that H2O2 and breaks it down. Phew, crisis averted.
Yeah.
But crucially, CK breaks H2O2 down into water and oxygen, O2.
Wait, doesn't GOX need oxygen for its initial reaction with glucose?
Precisely. The O2 produced by CTAT decomposing the toxic byproduct is instantly available right there inside the capsule to fuel the GOX reaction again.
Yeah.
So you get this amazing loop. GOX makes H2O2, CK destroys H2O2 and makes Q2, and that O2 immediately boosts the GOX reaction, producing even more protons. It tunes the waste product into extra fuel.
That's brilliant. It's like the system cleans its own exhaust and uses it as a turbocharger.
Kind of, yeah. It prevents the toxic buildup and significantly ramps up proton production, a self-reinforcing cascade.
Did they optimize the amounts? Like how much GOX versus CT?
They did. They experimented and found the sweet spot was a mass ratio of GOX-TET of about 1 to 0.5. That gave the best amplification and stability.
And the numbers back this up. How much better was the system at generating that proton power?
Significantly better. They measured the pH drop inside these nanocapsules. It went from a neutral 7.4 all the way down to 3.8. This huge pH difference translates into a really strong transmembrane proton gradient, a DH of about 1.4 units.
1.4. Okay, and how does that compare?
Well, systems using just GOX, without this catalase recycling loop, typically struggle to get much beyond a pH of maybe 0.5.
Wow. So nearly 3 times the proton motive force. That's a massive improvement. Not just tweaking, but a real leap in power output.
A stable leap, too, because of that internal cleanup crew.
Okay, so they've built this powerful self-clogging proton engine. Step 2 must be turning it into the full artificial mitochondrion. How did they assemble the final thing, the GC at SiO2 at ATPase?
Right, so they took that GC at SiO2 nanocapsule core we just talked about. Then they wrapped it in a lipid bilayer, a liposome. That's the lips part. This forms the membrane, just like in a real mitochondria, across which the proton gradient is established.
The boundary, okay. And the actual ATP making machine.
That's the third piece. They embedded ATPase enzymes into that lipid membrane. ATPase is the molecular motor that actually uses the flow of protons to synthesize ATP. And get this, they actually extracted the ATPase they used from fresh spinach leaves.
From spinach. I love that. So you've got the proton pump core inside, the membrane around it, and the spinach-derived ATPase motors embedded in the membrane, ready to go.
Ready to go. The strong proton gradient, that APH of about 1.4, pushes protons through the ATPase.
And is that gradient strong enough? Does the ATPase need a certain level to kick in?
It does. The threshold for these kinds of native ATPases is generally around a APH of 1.0. Their system consistently generated 1.3 plus or minus zero point 0.2 units, so comfortably above that threshold.
So plenty of power to turn the motors.
Exactly. And this tight coupling resulted in pretty impressive ATP production. They measured rates up to around 20 to 40 nanomoles per minute per milligram of the complex. Steady, continuous ATP synthesis fueled by glucose.
Okay, this is really coming together. They've built the power plant. But the ultimate test, right, is can it actually do something useful? Can it power other processes like a real mitochondrion does inside a cell?
That's the crucial question. Making ATP is great, but can you use that ATP to fuel something else complex? So they moved to the next level, integrating this whole artificial mitochondrion inside a model synthetic cell. They used giant unilamellar vesicles, GUVs.
GUVs, basically big, simple, cell-like bubbles made of a membrane.
Exactly. Perfect little contained environments. So they put their GC at SiO2 at Lips ATPase inside these GUVs.
The power plant is now inside the synthetic cell. What did they try to power with it?
They set up an ATP-dependent metabolic pathway inside the GUV, specifically a pathway to synthesize NADH. They added the necessary enzymes, hexokinase, G6PD, and the starting material, NAD plus chain. NADH is, a really central molecule in cellular metabolism involved in energy transfer.
Right, a key electron carrier. So if they can make NADH using the ATP generated inside the GUV by their artificial mitochondrion, it proves the whole system works together.
Precisely. It demonstrates metabolic coupling. The energy is generated and then immediately used locally to drive another complex process.
And did it work? What was the key result? Did putting it all inside the GUV matter?
It mattered hugely. They compared the NADH production in the GUVs, where everything was compartmentalized, to a control group where all the same components were just mixed freely in a test tube. The GUV system, with the encapsulated artificial mitochondria powering the NADH pathway inside, showed a 53.6% increase in NADH production compared to the free-floating system.
Wow. Over 50% better just by putting it all in the right place inside that vesicle structure.
Yeah, it really highlights the power of compartmentalization in biology, even in these synthetic systems. It makes reactions much more efficient.
So that confirms it then. They successfully mimicked the whole chain.
Yeah.
Glucose fuel, proton gradient ATP synthesis, powering a downstream metabolic pathway, all within a confined cell-like space.
Exactly. It effectively recapitulates key features of how mitochondria power cells, but in a completely engineered bottom-up system.
That really is a landmark achievement. Building a stable, self-fueling artificial mitochondrion that can run on simple glucose and autonomously power complex biochemistry inside a synthetic cell, that really feels like it cracks the energy problem for the field.
I think it's a major step forward. It provides a really effective and, importantly, versatile platform. You could potentially swap out the enzymes, tweak the outputs. It's a robust foundation. Beyond just building artificial cells, this gives us a fantastic new tool to study bioenergetics itself, how energy works in compartments, maybe even understanding mitochondrial diseases better down the line. It opens up a lot of possibilities.
A powerful new tool indeed. That's all we have time for in this deep dive. Thank you for unpacking that with us. And for everyone listening, please remember to like and subscribe. It really supports the Sans Science experience.